Hello and welcome. Today we're going to move on with the next chapter, which is about the molecular genetics. Now, when we say molecular genetics, then we're going to go through the genetic material that is found in all cells of our body and in all cells of the living organisms. So when we talk about the genetic material, it's basically the DNA. So molecular genetics means that the DNA is basically a molecule, and this was achieved after several experiments until they found that this is really a molecule. When I say molecule, I'm talking about several atoms that are connected together. So in this chapter, we're going to highlight the DNA, its shape and the structure, and what are the main components of the DNA and how does it carry the genes that we have in our body. So by starting with the DNA and the three initials, DNA stands for deoxyribonucleic acid. So there is a type of molecules that are found in our body that are considered as nucleic acids. And when I say these molecules are in our body, it's because we have also the carbohydrates, we have the fats, and we have the proteins. And those were the molecules of life that we have mentioned in chapter number 1. So in this chapter, we're going to move on with the fourth type of a molecule of life, which is basically the DNA. So DNA stands for deoxyribonucleic acid. So the group of molecules now that we are going to talk about are the nucleic acids. And when I say deoxyribose, that's because this molecule was one oxygen less than the ribose sugar that we will see in the other type of nucleic acid, which is RNA. So we have two types of molecules that are included in the nucleic acids, the DNA and the RNA. At first, we're going to highlight the DNA in the first part, and then in the second part, we're going to talk about the RNA and its role in our cells. So DNA is deoxyribonucleic acid, and the shape of the DNA is, as we all know, is the double helix shape. Now, how did they find that the DNA shape is this way? How did they find that the structure contains several types of atoms to make that shape? This was achieved by many scientists that worked hard to achieve that result. So we will start with the first two scientists, which were Hershey and Chase. Hershey and Chase are two scientists that proved that DNA moves and travels and it is transferred from one cell to the other. Now, how did they do that? Look at this picture. It involves the steps that they have made to achieve the result that they have obtained. They used a bacteriophage, which is basically a virus that attacks bacteria. And they have used radioactive material. When I say radioactive, that means it is shiny. So as you can see here, they have used radioactive sulfur to be included in the proteins. And here they have used the radioactive phosphorus that was involved inside the DNA. So in the first experiment, they have made the proteins radioactive. And in the second experiment, they have made the DNA radioactive. And they allowed this bacteriophage to infect the bacteria, as you can see. So this is the bacteria, and these are the bacteriophages that are infecting the bacteria. Now, they continued with the process. They made the blending, and they made then the centrifuge. And the centrifuge is basically a process by which we separate things from each other. Now, they have tracked where the radioactive phosphorus and where is the radioactive sulfur went. And they found that the radioactive sulfur was kept outside of the bacteria. It did not go through the bacteria or entered the bacteria. However, here, the radioactive phosphorus, they found that it was transferred from the virus into the bacteria. And by time, they found that the bacteria is now radioactive. What does that indicate? It indicates that this bacteriophage, as it attacks the bacteria, it injects a specific molecule inside the bacteria to allow the survival of the virus later on. Now, the injection of this material was basically the DNA of the virus and not the protein coat, because the viruses basically are made of protein coats and they have their DNA inside that protein coat. So by the end of this experiment, they found that what was transferred basically is the DNA or the genetic material that is found inside the virus and not the protein coat that was there. So in their experiment, they are basically proving that DNA and not proteins are the molecules that will be transferred when one bacteriophage attacks, for example, a bacteria. And this is, of course, will be applied for all the genetic material that is being transferred from one generation to the other. And it's not the proteins as we have seen in this experiment. The second two scientists were Watson and Crick. Watson and Crick were two scientists that worked a lot. As you can see, these two scientists are standing beside their model for the DNA. So they have found how the DNA is arranged with the atoms and the molecules so that the whole structure is made, and the double helix structure that we know is how they have made it using sticks and balls so that they can make the structure as a whole. Now, the model of the DNA is a double helix molecule, and the double helix means that we have two strands, and these two strands are let's say bending around each other and coiled around each other so that they can make this structure as we know it. The third scientists that were involved also in the discovery of the DNA were Michelson and Stahl. Michelson and Stahl also worked on how the replication of DNA is going to happen. Because we know that in each and every cell we have our genetic material, and if we want to make a new cell, like for example in growing or in repairing or in development, so we know that the DNA should be transferred to the next cells that are going to be there. So Michelson and Stahl made a little bit of a complex experiment so that they can find how does the DNA replicate. And in this experiment, as you can see, they had a media of nitrogen 15. When I say nitrogen 15, I'm talking about an isotope of nitrogen, because we all know that nitrogen in the periodic table is N14, which is basically the light nitrogen, we call it. They used the isotope of nitrogen N15 and they call it the heavy nitrogen. They made a media that is filled with nitrogen 15 only. Why did they choose nitrogen at this point? Because one of the basic atoms that is available in the DNA is the nitrogen. So they used the N15 now at first, and it was a media where the bacteria is growing. They allowed the bacteria to grow, and to grow for the bacteria, they need to take that nitrogen and use it in their replication and in their growth and in their making of new cells. So basically, after a few hours, we found that all the bacteria that is found in medium 15 had DNA that was considered as the heavy DNA, because the bacteria used all the N15 that was found in the medium. Now, after that, they prepared a media of N14. And N14, as we said, is the light nitrogen. And they transferred a little part of this media into N14. So what was the result at this point, which was a little bit different? The result was that the DNA that was formed inside the bacteria had both the heavy and the light nitrogen. So there is a little bit of suspicious now. How did that happen? Why do we have half of the DNA is N15 and N14, although we have a little bit of N15 now added to the N14? They continued their experiment by again taking part of these bacteria that had both the light and the heavy, and they transferred it again into N14 again. And they found that now we have a whole DNA of light nitrogen and a little bit of the DNA that has both the N14 and the N15. And they continued their experiment by adding also a little bit of this media into a media of N14 until they found that most of the DNA in the bacteria is N14. However, we still have a little part of N15 there. So in this experiment, they found that the replication of DNA is a semi-conservative replication. That means each two strands of DNA that are coiled around each other in a double helix shape, they are going to unwind and they're going to add new strands on both of their old strands. And this is how we can imagine that. If we're looking at, for example, the DNA with the heavy, that means these two strands are heavy and heavy. Now, if they want to use new nitrogen, they are going to use it from the N14 because they were added to this media. So at this point, the heavy N15 is going to use the N14 nitrogen. And the other strand of the heavy is going to use also N14 because this is the only nitrogen that is available in the media. At this point, we will find that the new DNA that was formed with an old strand that has N15 and a new strand that has N14. And the same goes for the other strand. So basically, all the DNA that we have at this point has both the old and the new, has both the N14 and the N15. But by time goes, the only available nitrogen is N14. So at this point, we are going to use the N14. So even if the strands of the N14 and 15 are using now new nitrogen, the only nitrogen that is available would be N14. So the N14 will take now new N14, and the N15 is going to take also N14. And by this, we will form again these two strands. And as we said, by time, more and more N14 is going to be engaged in the DNA strands rather than the strand that has the N15. Now we're going to dig deep into the DNA molecule and see what are the main constituents of the DNA and how does the structure form because of the molecules that are available inside it and because of the atoms and the bonds that they form. So by looking at the DNA deeper, we can see that the DNA molecule is a polymer, just as we said in the carbohydrates, the proteins, and the fats. A polymer means that a molecule that is made of many basic units, and these basic units are called monomers. So when we look at the DNA, which is a polymer, a large molecule, it is made of basic units. These basic units are called nucleotides. Nucleotides came from the word nucleic acids. So the monomer is the nucleotide and the polymer is the nucleic acid. Now, this nucleotide, which is one molecule, as you can see, it's a large molecule, it consists of three parts that we have to recognize. The first part that we should know, which is the deoxyribose sugar. This deoxyribose sugar is a pentose sugar. That means it has 5 carbons, and there is one oxygen that was removed from this part, and that's why we call it deoxyribose sugar. Ribose means that it has 5 carbons. This deoxyribose sugar is connected with one part with a phosphate group, and it is connected with another part with nitrogen base. This is the nitrogen that we have said earlier. It is contained in the DNA. So as you can see now, the nitrogen is available in here, and this is called a nitrogen base. So the basic component of DNA, which is the monomer, is called a nucleotide. It is composed of 3 parts. The first part is the deoxyribose sugar. The second part is the phosphate group. And the third part is the nitrogen base. However, the nitrogen base, they found that it is not always the same. Even this part of the nucleotide is changing. And this change, they found that we have 4 different nitrogen bases that are connected with the deoxyribose and the phosphate group. So we have to remember that this part of the DNA, which is the phosphate and the deoxyribose sugar, do not change in the nucleotide. However, this part, which is the nitrogen base, is changeable. That means we have 4 different structures that are connected to the deoxyribose sugar to make the whole DNA molecule. By looking now at the 4 different nitrogen bases that we know, we can see that we have these nitrogen bases that are 2 cycles. And when I say cycle, I'm talking about this hexagon and the pentagon that is connected to it. This is also a 2-cyclic nitrogen base. This is a 1-cyclic nitrogen base, and this is a 1-cyclic nitrogen base. This is as well and that 1 cycle, but we will discuss it later. So the 4 nitrogen bases that I have mentioned are the adenine, which we call it A usually, the guanine, which is G, the cytosine, which is C, and the thymine, which is T. Let's talk about uracil later because it is not in the DNA, it's in the RNA, which is the other genetic material that we will be discussing. So now, the 4 nitrogen bases are the adenine, guanine, cytosine, and thymine. And if we will make it easier, it's A, G, C, and T. Now, there's a little bit of differences, as you can see. We said that we have a 1-cyclic nitrogen base, and we have 2-cyclic nitrogen bases. The 2-cyclic nitrogen bases, which are the adenine and the guanine, are called purines. However, the 1-cyclic nitrogen bases are called pyrimidines. So these, they look like each other, except for the methyl group that is available in the thymine. These 2, they look like each other. However, we have a double bond O here instead of the amine group that is found in the adenine. So there's a slight differences between these nitrogen bases. However, they are the main components of the nucleotides that make up the DNA eventually. So we have A, G, C, and T. Now, by looking at these 4 different nitrogen bases and how they are connected, we have to remember that DNA is a double-stranded molecule. So it has 2 strands that are twisted, and they form like a twisted ladder, we call it. And this is the double helix molecule that we call the DNA with. How does that form? It's basically one of the pyrimidines is going to connect with one of the purines. So the adenine, which is a 2-cyclic nitrogen base, is going to connect itself with the thymine. So it's always A with a T. How can we remember that? It's apple on a tree. So A with a T at all times. The other 2-cyclic nitrogen base, which is the guanine, is going to connect itself with the cytosine. So the G is always connected with a C. How are we going to memorize it? By cargo. Cargo. So A with a T, C with a G, T with a A, G with a C. So this is how we're going to make our DNA. This is how our DNA is basically composed and how it is formed in the final structure that we will now see. So look at the nitrogen bases as you see in this picture. Look at the deoxyribose sugar, which is purple in color in here, and the phosphate groups that are green in color. And look how they do not change in the nitrogen bases, in the nucleotides that we are making or composing the DNA with. However, look at the adenine, which is a 2-cyclic molecule. It is connected with a thymine, which is a 1-cyclic. The cytosine is connected with a guanine. Guanine is connected with the cytosine. And if we see another thymine, it will be connected with the adenine as well. So at all times, the basic strands of the DNA, which are the 2 strands, are made of the deoxyribose sugar that is connected with a phosphate group, which is connected to the deoxyribose sugar with another phosphate group, another deoxyribose sugar, and so on. So the basic component of the 2 strands of DNA are the deoxyribose sugar and the phosphate, deoxyribose sugar and a phosphate. However, The only thing that is changing in the DNA is what connects these two strands together. And how they are connected, as you can see, we have slide dashes in here that are connecting the adenine with the thymine and the cytosine with the guanine as well. These dashes are very important because they represent the hydrogen bond. And the hydrogen bond was discussed in the water earlier in the first chapter, and it is very important in the making of the structure of the DNA as a whole to keep these two strands connected. So we have a double, or let's say two hydrogen bonds between the adenine and thymine. We have three hydrogen bonds between the cytosine and guanine, as you can see. These bonds are unchangeable. When we say unchangeable, that means we always have two hydrogen bonds between A and T and three hydrogen bonds between C and G. It's never the way, the reverse structure. So at all times, we don't see three hydrogen bonds here. We don't see two hydrogen bonds here. So at all times, apple on a tree, A with a T with two hydrogen bonds. C with a G, cargo, is always three hydrogen bonds. And this is what keeps basically the two strands of DNA intact together and forming the ladder or the twisted ladder that we have talked about. So when we look at the DNA, we can see that there are two strands and we have the lines in between. And these lines are basically the hydrogen bonds that are connected in here. Now, if we're talking about the percentage of each nitrogen base, we can see that the amount of adenine should always be equal to the amount of thymine. Because as we have discussed, always A binds with the T. So however we have A, we will have T in front of it. However, for the C and G, it goes the same. The amount or the percentage of cytosine will always equal the percentage of guanine. And the four percentages of these four nitrogen bases would always be 100%. There's a little bit of an example. When we say, for example, a DNA molecule contains 20% of it as cytosine, and they asked for the percentage of adenine. So when I say cytosine is 20%, that means guanine is also 20%. So both of them is 40%. And what is left now for the A and T is 60% of the 100. So the 60 will be divided now equally between the adenine and the thymine. So basically the DNA molecule now, we can see how it is two strands that are coiled around each other, and we have those lines which are the hydrogen bonds that are connecting these two strands, and they are made because we have nitrogen bases that are on both sides. This DNA is coiled and supercoiled around histones of proteins to form what we know as the chromosome. Where are our chromosomes? They're inside the nucleolus, which is inside the nucleus. So now you can imagine how the DNA molecule is very tiny and very small. However, it carries all the genetic material that we know. In this video, we're going to show you how the DNA molecule is found inside our nucleus, and the structure of DNA is very important. Here's a look of our cells and the cell organelles. We know that we have a cell membrane, we have a cytoplasm, we have the mitochondria for the energy, and we have the vacuoles that contains all the water and the minerals. We have the Golgi body to deliver things in, and we have the nucleus, which is the largest one. We're going now to enter inside the nucleus, and we can see now this is the chromatin, which is basically the DNA that is coiled around histones. These histones are proteins basically, and this is how the DNA double helix is made. Genes are basically parts on our DNA. As you can see, it is highlighted. So the whole DNA, as we will mention, that it is basically 11 meters length. And in our body, we can see that all the DNA is made of the two strands, the backbones we call them. These are the sugar phosphate backbone. And we have the nitrogen bases, as you can see. it's always A with a T, and you can see that we have the G with the C. We will discuss the purines and the pyrimidines, which are the names of the nitrogen bases. The nucleotide, as you can see, is the basic component of the DNA. It is the monomer that makes the whole DNA molecule as the double helix molecule. So it is basically a molecule that we know. Thank you.